Good morning. It's good to see everyone. Welcome to Cardiology Grand Rounds this morning. This is our Locke Endowed Visiting Professor Grand Rounds. And I just want to acknowledge and thank the Locke family for making this possible. John Locke Jr. was a victim of heart disease, and at the time of his death in 1968, Mr. Locke bequeathed his estate to support the care, treatment, and research of heart disease. Upon the recommendation of his personal physician, Dr. William E. Watts, funds from his estate were used to establish a number of programs in the Seattle area, including the John L. Locke Jr. Coronary Care Center at Swedish Hospital and the John L. Locke Jr. Health Sciences Computer Center here at the University of Washington School of Medicine. In 1979, Irene Fisher Locke, mother of John L. Locke Jr., created the John L. Locke Jr. Charitable Trust with a personal gift. And income from the trust was to be used to enhance in perpetuity the programs in heart research and treatment established by John Jr.'s bequest. This set of funds funds the Family Endowed Chair in Cardiovascular Research and Treatment at the University of Washington, research projects in the Division of Cardiology, and of course, our Locke Visiting Professor series. And I am especially happy to be able to say a big welcome to Tom Coppola to the visiting lectureship. Tom graduated a while back from Princeton University in undergrad. He then went to Harvard Medical School and then Johns Hopkins for public health. He did his internship and residency at Brigham and Women's Hospital, followed by a clinical and research fellowship at Johns Hopkins, heart failure and transplant also at Hopkins, research fellowship at the NHLBI, and even an honorary senior visiting fellow a little bit later in his career at Cambridge. He spent a year as an assistant professor at Hopkins before he landed at the University of Pennsylvania, where he's been ever since, and has risen through the ranks incredibly quickly to assume the full professorship, the Herbert C. Rohr Professor in Medical Sciences, and the chiefdom of the Division of Cardiovascular Medicine. Along the way, he's been the Director of Heart Failure Clinical Research and Clinical and Translational Research Center. Tom has published in many different articles, in many different highly respected journals, including Nature, Circulation, Cell, JAMA, and the New England Journal of Medicine, and despite his current role, he continues to be extremely active in research and publishing. His list of grants is longer than most of our CVs, and most notably, he has multiple connections with the NHLBI. He has received the Presidential Early Career Award for Scientists and Engineers, a highly prestigious award, and he's been elected to the American Society for Clinical Investigation. And more than that, to be perfectly honest, Tom is just a great guy. One of the nicest people that you'll ever meet. He is an incredible mentor. His list of mentees is maybe even a little bit longer than his list of grants. And he has contributed greatly to the careers of many, many people. And it was a great thing when actually he was appointed chief at Penn. There was much rejoicing when that occurred. And he's done a fabulous job since then. But we're very lucky to have him give a talk on his expertise in genomics of heart failure. Please welcome Tom Coppola.
Thank you, Jim, for that incredibly kind introduction. I hope this lecture lives up to those expectations, despite the incentive I hear you come to watch the lecture. It's a real honor for me to be here, and I really want to thank the Locke family for making these events possible. It's very important to have philanthropy support academic enterprises and innovation. We really can't do new things without it, so I'm very honored to be here, and I want to thank Rob and Jim for the kind invitation. I had a great day yesterday meeting with many of you. So what I wanted to do today was first I'll show you a picture of where we are. Whenever I go to a different division, I kind of reflect on our own division. And they're all different. They all have different strengths. And our natural strength is sort of pictured by this physical structure here. So this is our Smilo Center for Translational Research. This is where our basic laboratories are. This is a very big floor. And right here is the Division of Cardiovascular Medicine, where all the clinical leaders and clinical programs are, and the fellows and trainees. And right down here is our outpatient center. The hospital is over here. So we're very positioned to take science and bring it into the practices and take things we learn from the practices to inform the science. And that's been sort of the theme of my career of translational medicine. And what I wanted to do today was to give you an overview of one enabling technology, genome science, and how that's impacted the field of heart failure medicine. I want to take you through some of the general things that have happened in the field over the last 2 decades and highlight the work of many people, including our own work, and hopefully you'll learn something. Couple disclosures. So this is heart failure. So I'm a heart failure transplant doctor, and this is a very simple model of heart failure. It's the cardiac response to injury, right? So you're born with a heart, you are exposed to various injuries over the course of your lifetime, and these can be very varied, and you're all familiar with them. Valve disease, coronary disease, hypertension, and inherited cardiomyopathy mutation. The heart remodels, and that over time causes the syndrome of heart failure that you all treat in the office, fluid retention, shortness of breath, etc., and it propagates. I would submit to you that each of these different processes is influenced by our inheritance. We inherit genetic variation from our parents, and those in concept could affect each of these processes. So that's the task, is trying to understand that. Well, what's the evidence that heart failure is an inherited syndrome? There's actually very strong evidence, and you may or may not be familiar with this. This is an older study from the Framingham Heart Study, in which the question was asked, if you considered family structures in Framingham, and there's parental heart failure, and you adjust for exposures, what's the odds of having a child with heart failure? It turns out that if both of your parents have heart failure, just broadly defined, you are twice as likely to have heart failure. So there's substantial heritability there. And a more recent iteration of this, which was perhaps a little more rigorous, is a Swedish national study. This was really interesting. So Sweden has a lot of data about the health of the population and their specific conditions. In this study, adoptees were studied, several 100,000 of people who were adopted, and then went on to get heart failure. And what they asked was, among those people who developed heart failure in their adopted family where they were raised, what was driving the risk? Was it the adopted family's risk of heart failure, or was it the biological family's risk of heart failure, who they never saw because they were adopted? They still knew who they were. And the answer was about 30% of the heritability was genetic. So there is a clear inherited predisposition to heart failure. And importantly, this is true even if you exclude primary cardiomyopathy, HCM, DCM. So there's clearly a genetic basis for the syndrome that we're talking about. Okay, well that's good. So is there value in pursuing this as an endeavor? So it's inherited. Why should we take on the big task of trying to define the genetic basis of heart failure and its predisposition? And I'll skip to the take-home message. This is the one thing I want you to leave with related to heart failure. There is already value. So we already have rare heart failure variants, a whole catalog of them, that are essentially causal for heart failure. They're clinically actionable. And in fact, the American College of Medical Genetics and Genomics has a list of genes that if you find a variant in them, you should recontact a patient in the course of a research study. Half of them relate to heart disease, and most of them are cardiomyopathy. So there's real value here already. And one of the points I want to drill home is that I think as cardiologists, not at a place like UW, but nationally, we under-leverage this data in the care of our patients, and that's a challenge for the field. We'll come back to that later. So rare heart failure variants have value already. Common heart failure variants with smaller effect, a little more controversial. This is your 23andMe, right? You get your genome scan of common variants. Over time, the value of this approach in clinical medicine is starting to become more apparent, and that relates to polygenic risk. We'll come back to that too. Now, both of these types of information can inform our understanding of heart failure mechanisms and drug development, and that's the other benefit there. Okay, so that's the one thing I want you to remember. We'll come back to this at the end. And now let's talk about how to actually do this. So how do we take what we know is an inherited condition that's really complicated and really start to dissect it and understand it? And this has been the work of many in a whole field. And I would boil it down to 3 challenges. There are 3 challenges that pertain to heart failure. One is the phenotype. The second is understanding the mechanism for each of the findings. And the third is how to use that to impact patient care. So I'm going to, for the rest of the talk, take each of these challenges and outline how the field has approached it and what we've learned along the way. So the phenotype. Heart failure is complicated. There are many complex traits in medicine and in biology. What's different about heart failure is not only is it a complex trait, it's very heterogeneous. So if you think about an LDL level, that's a complex trait, right? We've learned a lot about the genomics of cholesterol metabolism. It's complex, but it's very specific. It's very quantifiable, and that makes it very tractable for genetic analysis. Heart failure is the opposite. It's murky, it's distal, it's almost an 18th century phenotype, right? There are multiple subtypes, multiple etiologies, so we need to get our hands around how to do proper genetic studies in a heterogeneous murky trait, okay? So how do we do that? Well, the first is to start where it is the least heterogeneous, and that is in primary cardiomyopathy. And this is all familiar to you. Classic dilated cardiomyopathy, hypertrophic cardiomyopathy, ARVC, non-compaction cardiomyopathy. I would add, as you're thinking about this, amyloid is important to think about too, because that's now quite an actionable genetic disease. And we've learned a lot about these over the last 30 years, and this is work done by many people through linkage analysis. Clear Mendelian disease, where there's a trait, a disease trait that sorts in a family. There are many approaches to try to find variation in the genome that segregates with the disease as it's passed down through a family, and we've learned a lot about this. And this is what is the basis of our genetic testing profile now. There's overlap amongst these syndromes, but there's a panel of genes with mutations, causal mutations, and we have to think about this. We have to think about this when we see patients. Many of the patients you see with so-called idiopathic dilated cardiomyopathy, a substantial portion of it, anywhere from 30 to 50%, is probably genetic in origin, at least in part. And we need to get used to that idea and have it impact our practice. Titin is a very interesting example. So this is, again, over the last 12 years, Titin is an exception. It's the biggest gene in the body, and truncation mutations in Titin have been strongly associated with so-called idiopathic dilated cardiomyopathy. So I was talking to Chuck Murray about this yesterday. He's got some really interesting findings related to Titin. It's a very important aspect of cardiac biology. So Titin is the longest gene of the genome. It forms the backbone of the sarcomere. It performs a recoil structure. It's involved as a scaffold for sarcomere assembly. And through a Herculean sequencing effort, Dan Herman and the Sybins and many others identified that mutations that truncate Titin are a substantial risk factor for dilated cardiomyopathy. There's nuance to this, and it's becoming more and more interesting over time. One nuance is there are a lot of people in the population who don't have cardiomyopathy who harbor one of these mutations. And the answer there is pretty interesting. It turns out that the gene doesn't use all of its exons. Lots of exons are skipped and never expressed. It's the biggest gene in the body, so it can afford to skip a few exons. And those are the ones in which people in the normal population have a truncation mutation and don't have disease. So that's been another interesting nuance. But this is now on the genetic testing panels and can get tricky. But what's becoming more interesting is that Titin is at the root of other conditions that we don't normally think of as genetic. So here's an example from my colleague Zolta Rani. He became interested in peripartum cardiomyopathy. And how many people have seen peripartum cardiomyopathy? Pretty bad, right? And there are all sorts of theories about why this happens. There's a hormonal hypothesis, there's a hemodynamic hypothesis, and there's truth to all of them. But this was the first study to show that there's an inherited predisposition to peripartum cardiomyopathy. What Zoltarani did was collect as many cases across the world as he could find and sequence all their truncation variants in peripartum cardiomyopathy and compare them to controls using the same cardiomyopathy panel we use in genetic testing. And what he found was that Titin truncations were again substantially enriched in the peripartum cardiomyopathy patients compared to controls, so much so that it pretty much looked like dilated cardiomyopathy. So the working model is you inherit a Titin truncation mutation, and then there's a second hit. In this case, it's pregnancy, and you can develop a cardiomyopathy, right? And that two-hit model is now it's popping up in other places. Alcoholic cardiomyopathy. Very nice study by James Ware and Stuart Cook. What we call alcoholic cardiomyopathy also shows the same phenomenon. Patients who develop this phenotype are more likely to have Titin truncation mutations. So again, you have your Titin truncation mutation, you get a stress, in this case too much alcohol, and you get a cardiomyopathy. Chemotherapy-induced cardiomyopathy. There's a paper by the Simons again earlier this year in Circulation showing the same thing. So there's this concept of a titinopathy where the expression of cardiomyopathy can be traced back to a genetic risk and a secondary exposure. All right, so I think we'll probably see more of this over time, but this has become a very interesting nuance in the genomic testing panels for cardiomyopathy. Okay, so that's one way to start, which is to focus on clear Mendelian or extreme disease. Well, what about more common forms of heart failure? How do we tackle this? You know, the heart failure you see every day in the office. It's not so clear that it's an inherited cardiomyopathy. And the answer here is to go big. That's how you deal with it. One way to deal with some heterogeneity. Go really big, get as many patients as you can, and try to overcome heterogeneity with sample size. And that's what a GWAS does. And you've heard many GWAS presentations, I'm sure, by now. The simple idea is you collect as many cases as you can, scan their genome with whatever technology. It can be SNP genotyping, can be sequencing, and compare the allele frequencies in the cases with an unaffected population. And the areas of the genome where the frequencies are different can implicate genes that affect disease risk. And that's what you see here. And this has been done many, many times over the last 15 years. This is actually outdated. I believe now there are around 70,000 variant trait associations through GWAS. So this has been a large global enterprise. So when I came to Penn, as Jim articulated, this technology was coming online, and we were all very excited to use it for heart failure because maybe we'll learn something. So we started to collect our patients. You know, we formed cohorts, and I wasn't the only one. Started a study called the Penn Heart Failure Study and eventually grew into an NHLBI cohort at 3 sites of advanced heart failure referred to a subspecialty center, and we used it as a platform for GWAS, for biomarker studies, et cetera. And we actually learned some things. We found one convincing genetic variant that was real and has stood the test of time, okay? And this was like, I don't know, 7 or 8 years of work. We did a lot of all the other stuff along the way, but this has been replicated many, many times. We said, well, that's good, but there's got to be more than that. So what can we do and what are we doing wrong? And here's what we were doing wrong. We weren't big enough. So this is a timeline of genetic association studies for heart failure. Started off in the early 2000s in the candidate gene era where we'd pick our favorite gene and sequence that in cases and sequence the same in controls, and it was riddled with false positives because there was so much bias. So many of these have never been replicated and sort of died out. And then what I just described to you was sort of the moderate size GWAS era. And we found a few things. So did Nick Smith and Ben Meter, Eric Villard. And we said, all right, well, we're making progress. How do we really get the answer here? And the answer is go back to math and realize you don't have the power to really answer the question. You need more patients. And if you look at what was happening at the field in parallel, heart failure had been notably missing from these large GWAS consortia. I mean, look at the sample sizes. AFib consortium, 65,000 cases, 500,000 controls, blood pressure, coronary disease, et cetera. Where's heart failure? So we got together and said, this is going to be a lot of work. This is going to be a lot of time on the phone, trying to wrangle people to put their data together. I hope somebody emerges from the field to actually make that effort, because it sure as heck doesn't need to be me. And a person has emerged. His name is Tom Lumbers. He's a junior faculty member at University College London. And he has taken the effort of getting all of us together to collaborate and put all our data in one place. And we now have a consortium called HERMES, Heart Failure Molecular Epidemiology for Therapeutic Targets. So we finally have a proper global case control consortium for heart failure. And none of this is published yet, but it should be soon. Here's the first readout, and this is just simple heart failure, cases versus controls. We're now getting reasonable sample sizes, 47,000 people, many, many controls. I think we have 300,000 controls. And what we're learning is what you'd expect and what you wouldn't expect. So in all-cause heart failure, some of the strongest signals relate to AFib, coronary disease, and dyslipidemia. Well, tell me something I don't already know. AFib causes heart failure, right? Coronary disease causes heart failure. Dyslipidemia causes MI, which causes heart failure. But it's a good positive control that we're able to find that genetic risks for heart failure's precursors do in fact lead to clinical heart failure. And there's a causality here. The variation for PITX2, for example, that causes AFib. You inherit that before you get AFib or heart failure. So it's indirect evidence to support that AFib is a causal factor for heart failure, for example. I'm sure the electrophysiologists in the room will find that very, very pleasing to be able to find another rationale to ablate and treat AFib. Okay. In addition, though, there are new signals emerging, and this is where the new biology is becoming interesting. Things we hadn't considered before, which now seem to be real. And I'm not going to get into them yet because we want to make sure they're correct before we talk about them. So what I want you to take home from here is that there will be data emerging because we now have proper power to answer the question, what are the common variants that predispose to heart failure? So there'll be more coming on it. Okay, so that's one way to tackle a very large and heterogeneous phenotype. Just get as many patients as you can together. Brute force. And brute force works, but it has its limits too. Can we be more intelligent than that? I mentioned that heart failure is heterogeneous, but there are subtypes we're familiar with, right? I don't think we're good at it yet, but we at least have some basic subtypes. Ischemic or non-ischemic heart failure, right? That's a normal, typical divide in a heart failure study. So this is a study from the UK Biobank performed by Steve Lubitz's group. How many people here are familiar with the UK Biobank? All right, good. So for those of you who don't know, the UK made a major investment in population science about 10 years ago, where they enrolled 500,000 people prospectively in various regions of the UK. They've all donated DNA. They've been phenotyped using surveys, clinical records, and in many cases, measures, blood samples, EKGs, etc. But what's different is they've decided to make all the data available to any qualified investigator in the world. So you can apply and download these data and do an analysis. And that's what Steve did. And he asked a very simple question. If you just look at non-ischemic heart failure in the absence of valve disease and the absence of coronary disease, do you learn anything new? If you just look in that subtype. So he identified there are about 6,000 cases of incident heart failure in that 500,000 people followed so far in UK Biobank. And he looked at all 6,000. And then he looked at the subgroup of those 6,000, about 2,000, that have clear non-ischemic heart failure. And what he found was that you see different signals, right? So this is what we saw in the previous slide. PITX2, AFib locus, CDKN2B, a coronary disease locus. That's for all-cause heart failure. But when you look in just the non-ischemic heart failure, you start to see different signals. PITX2 goes down, BAG3 goes up. This is a known cardiomyopathy gene. And there's my old friend, that we found in the Penn heart failure study 10 years ago. So I was pleased by that. But the point is that we need to do a better job of parsing this phenotype into more homogeneous subgroups that have real biologic meaning. That's another way to tackle this complex trait. And this proves that. So there's a lot of effort now on what we call refining the heart failure phenotype, or heart failure phenomapping, or heart failure phenomics. And if I were to advise a trainee on how to get into this field right now, this is where I would start. Because the genetics and the genetic association is not hard anymore. The data are getting cheaper and cheaper to get. The analysis pipelines are there. What is the limiting factor is phenotyping. And for anyone who's in the heart failure field, you kind of know this in your heart. What is HFpEF anyway? How many HFpEF people are in the room here? So I collaborate with Julio Chirinos, who you may or may not know. And he's a HFpEF expert, and he focuses on ventricular vascular interactions and hypertensive heart disease. And I said, I believe in all that. But then you look at the trials and what we've called HFpEF, and it's anything from, funny little echo finding in an older person, to a true disease of hypertensive heart disease, to basically symptomatic obesity. And to think that there's a common genetic basis for that hugely heterogeneous population is really not a wise approach. So I think we need to do a better job at parsing these phenotypes into little buckets that are more homogeneous. Okay, so that's phenomics and phenomap. So we've tried over the years to do this with different approaches. So one approach you can imagine is a biomarker panel. So you can measure different biological processes in living human subjects by quantifying biomarkers in the circulation. And there are people whose BNPs are up and BNPs are down, and people whose troponins are up and troponins are down. And you can do this along many different axes. Inflammation. So BNP is inflammation. BNP is, you're familiar with, that's cardiac stress. SFLT is a marker of vascular remodeling. HSCRP is inflammation. ST2 is another marker of cardiac stress. Troponin I you're familiar with, which is a marker of ischemic damage, renal dysfunction, et cetera. So can you put all these together in a panel and describe and identify different subgroups of heart failure that behave differently? And the answer is you can. So based on these biomarkers alone, we were able to identify different subgroups that behave very differently. Look at the prognosis in this one compared to this one, a 15-fold difference. Now you can then ask the question, well, is there a common genetic predisposition that leads you down this pathway versus this pathway? So that's the kind of work that I think needs to be done. And people are starting to do it. It requires a lot of scale, bigger data sets, and many more traits measured besides just this biomarker panel of 7. So Sanjeev Shah published this paper now, I guess 4 years ago, which did the same thing in HFpEF, using echo features, echo traits. It's the same concept. Take a murky phenotype, use a lot of data to ask, are there subgroups in here that might be more meaningful? So I think this approach, phenomapping and phenomics, is going to be important as we try to parse this phenotype. Okay, so that's heart failure as a clinical trait, as a clinical syndrome. But we know that when you inherit a mutation or a variant, you just don't get heart failure. There are a lot of things along the way that happen first, right? And this is pathogenesis. So everything I've talked to you so far is pretty much asked, what are the gene variants that associate with a clinical flavor of heart failure? But how it actually works is you inherit a gene variant, and then stuff happens before you get heart failure, and then you get heart failure. You get a low EF. You get a change in your EKG, such as LVH, or a QT prolongation. Even more proximate than that, you inherit a gene variation, and maybe that influences which genes are expressed in your heart itself, and then that in turn leads to cardiomyopathy and heart failure. Well, you can quantify each of these traits using any of a variety of approaches, and now you've turned a murky clinical phenotype into a quantitative preclinical phenotype that's very tractable for genetic analysis. The analogy is MI versus cholesterol, right? You don't go from, well, you can go from genetic variant to MI, but many of the paths along the way go through the quantitative trait of a cholesterol level. These are the cholesterol levels of heart failure, these intermediate traits. A lot of good work has been done in mapping the genetic influences on these preclinical heart failure traits, and some of them are quite interesting, and I've listed some of them here. So I'll walk you through a few examples. Echo. We've got tremendous echo expertise at this institution. Are you guys part of EchoGen? Do you know?
Yeah, through charge.
Through charge. Okay. So EchoGen assembled a population of around, I think it's up to 40,000 echocardiograms and genomic data sets from populations across the world, and asked the question, which genetic variants influence cardiac mass, LV dimension, ejection fraction, etc. And they found things. And these are kind of interesting. So here's an example that I've been chasing. Left ventricular diastolic dimension. I'm a HFrEF person. You could probably tell from my prior HFpEF comments. And so this is a very interesting trait for me. And several loci have been implicated as influencing LV dimension, the diastolic dimension. MTSS1 is one of them. I'm going to come back to this later. So that's one example. This gives you a window into which genes might be relevant. Here's a very interesting example. And it's great to see Nona in the audience. She's done a lot of work on genetic influences on the electrocardiogram. The EKG, as it turns out, is a very rich set of many traits that you can conveniently get in large populations, right? Here's just one example. This is LV mass, good old LV mass. And I was surprised by this. I said, the EKG, that's early 20th century, right? Turns out it's very quantifiable. So this is 50,000 people and a meta-analysis done by my colleague Pim van der Harst. And he asked the question, what are the genetic variants that influence LV mass by EKG, by standard clinical definitions, Sokolov definition, QRS duration, etc. He found 50 genome-wide significant loci that influence LV mass. That's pretty cool. Which of these, and this gives you a starting point to think about the biology of cardiac hypertrophy in human subjects. So that I thought was pretty impressive. Okay, and this gets to work that we've been doing over the past, I'd say, 8 years, and that is, it turns out that which genes get expressed in the heart itself are traits, right? So you can quantify the expression level of a gene in the myocardium. Every gene is regulated by enhancers, promoters, and there are variants within those enhancers and promoters which can influence expression of those genes. And this is a nice review. I realized this was you, Rob, who wrote this review. Who would have thought? So this is called eQTL mapping, expression quantitative trait mapping. So what you see here is a gene, and its expression is regulated by an enhancer or a promoter. And you can imagine, what if you have a variant in there that fusses with a transcription factor binding site? What will happen to the level of those genes? Well, it turns out it can be potently affected by variation. Here's an example of a gene, just one variant, an expression of a gene ERAP2. This is in the heart. If you inherit the minor allele, if you have 1 or 2 copies of the minor allele, you have lower expression of this gene, and this is inherited, right? So you have an inherited reduction in this gene. That's kind of cool. So we decided, in a bit of a quixotic adventure, to map all these in the human heart in a consortium that we called MAGNET, Myocardial Applied Genomics Network. You got to have some kind of acronym in this business, or they don't take you that seriously. So this was an NHLBI consortium. I'm very fortunate to be a collaborator with Ken Margulies, who's probably amassed one of the world's largest myocardial biobanks over the past 25 years. And we have used this, along with collaborators from Stanford and Cleveland Clinic, to form a big biobank and do proper eQTL mapping. I won't walk you through all the work that's come out of this, but I'll just show you this one figure, which is we found way more than 1,000 genes in the left ventricle that are regulated by genetic variation. So this is a lot of data. Now, in parallel, there have been other efforts in the genome science community. This has been done in every tissue in the body now through a consortium called GTEx. So you can literally go to the web and ask, all right, I have a variant of interest. Was it due to expression of any gene in the body in any tissue? And just look it up. It's pretty powerful. So we're now left with a lot of information, right? And the information will keep coming as the analyses get easier, the populations get bigger. And the question, regardless of the phenotype, is what does it mean? This is the second question. How do any of these things lead to heart failure, and do we really care? And in the back of our minds, what we're kind of looking for is the PCSK9 story, right? This is sort of the holy grail of cardiovascular genomics. Loss of function variants in PCSK9 in populations dramatically lower LDL cholesterol. That was shown to be due to a change in LDL receptor recycling, which can be mimicked by an immunotherapy that interferes with PCSK9. And how many people prescribe these now? A couple of you, right? Yeah. They're a little expensive. But this was a very rapid drug development cycle. So in the back of our minds, this is sort of what we're all hoping for. But key to this is understanding how any given genetic variant acts. What does it do biologically? What's the mechanism? And the answer is, this is a lot of work. There's no quick answer to this. You know, each of these loci has to be dissected by people with phenotype and basic science expertise to really understand what's going on. I'm going to give you one example of many, just to give you a flavor for this. And this is work around that gene I mentioned, MTSS1, which was found in EchoGen to influence the size of left ventricles, okay? And this is work done by Xiao Wang, Mike Morley, in my lab, Kiran Musunuru in collaboration, Jeff Brandemardo, Ken Margulies, and Patrick Eleanor at the MGH. Another corollary here is keep in touch with your friends. So Patrick and I were residents together. Nolan and I were med school together. And Kiran and I met when I was a fellow. It's amazing how small a community it actually is when you get down to it. You can call your friends up to help you out. So this is that figure I showed you. This is MTSS1, and this is LV diastolic dimension in around 40,000 people. There's a significant association between variance at this locus and LV diastolic dimension. Okay, that's interesting. So now what? The reason we got interested in this is that very same variant in that prior figure, when we looked in our eQTL map, had a strong association with expression of MTSS1 in the left ventricle. So if you inherit the minor allele of this gene, of this SNP, okay, you have lower MTSS1 levels in your left ventricle. Well, that's good. Well, how's that working? Well, we fine map this region. Here's the gene, MTSS1. This is the position on chromosome 8. This is the variant, RS125415595. And we asked, well, where is this thing? And these little dots here are the association strength between that variant and expression of MTSS1. And you can see that this SNP is right at the peak, and it sits right in a predicted enhancer. So we say, maybe this variant somehow messes with that enhancer, and that's the mechanism through which it lowers MTSS1 expression. Okay, so we tested that. I called up Patrick and I said, Patrick, you've got this zebrafish thing going. I'm not a zebrafish guy. How many people here are zebrafish people? Okay, good. Then don't criticize me. This is a zebrafish, developing zebrafish. And what this assay is meant to show is where is this enhancer active. So we cloned this enhancer region. We cloned a very small region right here. We put it upstream of green fluorescent protein. You inject it into zebrafish embryos, and wherever that enhancer is active will light up green. And so we then said, well, what's green? And what's green is the heart and developing muscle cells. This is like the yolk sac, so that's not relevant, it goes away. So we said, aha, this is a cardiac and muscle-specific enhancer. Well, that's getting even more interesting. And then here's the heart. You can dissect the heart out of a zebrafish. It's a little different. This is the ventricular chamber, this is the atrial chamber, and this is the outflow tract. And green is enhancer activity. You could see activity is mainly in the ventricle, not in the atrium or the outflow tract. We said, well, that's getting more interesting. So this looks like a cardiac-specific enhancer, which would make sense with the observational genetic data we had. So then we spent some time convincing ourselves that this was a causal relationship. So we got rid of the enhancer and showed that expression goes away in a cell model. We then tried to figure out what was happening at the genetic level. So here in neonatal rat cells and in another cell line, we cloned the two haplotypes. So our hypothesis is that if you inherit the minor allele, you'll have lower levels of MTSS1. So you should have lower enhancer activity if that's what's driving it, right? And that's what we found. If you inherit the minor haplotype here in green, you have lower enhancer activity than if you have the major haplotype. So all of this is a consistent story that if you inherit this variant and it's the minor allele, you have lower MTSS1, and in turn, in the populations, you seem to have a smaller left ventricle. So we'll come back to that in a minute. Okay, so then we said, well, let's really go back and scrutinize many populations. So we looked at this same variant, and we asked, all right, what does the minor allele do? The MTSS1 lowering allele. We went back to our cardiomyopathy case control studies, which again are moderate in size. And if you inherit the minor allele and lower MTSS1, you have a lower odds of cardiomyopathy. That's pretty cool. What about LV mass by EKG? I showed you how powerful that was. Well, if you inherit the minor allele, you have lower LV mass by EKG. You have lower LV mass by echo. You have less ventricular dilatation by echo. And you have an increased fractional shortening by echo. So this is telling a consistent story across these various traits. The MTSS1 lowering allele seems to show a cardioprotective type phenotype, right? Lower mass, smaller hearts, higher ejection fraction. What's especially interesting to us, though, is that it's the loss of function that creates protection. And in biology, we're good at breaking things. We're not as good as making things. That's the PCSK9 story. Get rid of it, and cholesterol's lower, and that's protective. So, we got a little excited and said, well, maybe someday if we figure this out, this could be a therapy by ablating MTSS1 in the heart and protecting you against adverse remodeling. That's a big stretch, but it's a stretch that the NIH bought. So we have a lot of mouse work to do over the next few years. Here's our first attempt at getting more in vivo on this. So we knocked out MTSS1 in a mouse model. And in fact, this is a global knockout. When you knock out this gene, you do have smaller hearts in these mice. The ejection fraction is slightly bigger. Fractional shortening is slightly more, et cetera, et cetera. So it's all a consistent story. And we just published this last month in a brief report. We have a lot of work to do, though. I don't know if this will ever lead to anything, because we still don't, this is leading me down a path that is very unfamiliar. So first of all, the name of this gene is pretty inauspicious. Metastasis suppressor 1. It doesn't sound like something you want to ablate. So what is this gene? Well, it was first described in bladder cancer cell lines as a marker of metastatic potential through a gene expression scan. What's known about the biology of it is that it is in the IBAR family, which is a family involved in actin cytoskeleton assembly and the formation of filopodia on membranes and intercellular connections. It has nothing to do, as far as we know, or as far as anyone knows, with sarcomeric function. So it could be that this is a tissue phenotype, that if you get rid of this, there's something impaired about tissue level remodeling, and that limits tissue level remodeling. That's as far as we've gotten, or I've gotten in my understanding of how this might work. So we have a lot of work to do, and lots of friends to call up who are cell biologists. Okay, so that's just one example of the hundreds of loci, of how to dissect one of the hundreds of loci that have come out of these studies, and it's a lot of work. So maybe there's a way to do this in high throughput parallel fashion. If anyone's going to figure it out, it's probably Jason Doerr. But for now, we just have to tackle these things one at a time. Okay, so that's the science. What about helping our patients? So how can we bring all this burgeoning information to care and to really help our patients? And the answer we've come up with is to build centers that combine science and care in one physical environment. And we are positioned for that based on our physical structure. So this is a center that we started about 4 years ago called the Penn Center for Hereditary Cardiovascular Disease. And what we've tried to do here is take all the dispersed elements of medical genetics and bring it into the cardiovascular division and the cardiovascular clinic, so there would be one structure. We have a medical director, Anjali Owens, who's a phenomenal advanced heart failure trained specialist who also has run our familial cardiomyopathy clinic for many years. And we have a scientific director, Kiran Musunuru, who's a very creative genome scientist and cardiologist. And I said, why don't you guys work together to build out a center that brings these new advances to patients as much as possible? And they've done a great job. We've recruited more people. Charlene Day is going to come and join us in July, along with some of our trainees. And I want to just emphasize to the trainees in the room, if there are any, that this is only going to grow. And I'll give you an example. We have a big health system across the street from us, Jefferson, and they got the great idea that they're going to offer all their employees free genetic genome scanning through color genomics. You know, it's like 23andMe. And they did this, and several of their employees were alarmed to find out that they might have cardiomyopathy genes, cardiomyopathy variations. So they called up the leadership at Jefferson and said, where do we go? And they said, well, we don't do genetic counseling in cardiovascular disease. We just do cancer. So I don't know. So they Googled around and they found us. And now we're getting referrals from there. People are coming with their alarming stories about what they're learning from 23andMe and unable to interpret it because it's a moving target. What's actionable, what isn't, changes over time. So I think that over the next 10 years, like it or not, cardiovascular practitioners are going to have to be comfortable with a genomic data set in thinking about patient care. And I think the way to do it is to get more genetics counselors and more expertise and more trainees who can fill these roles in cardiovascular programs. So that's a long way of saying, train up in this because we want to hire you. Okay, so they've done a great job at increasing the referral base. They've expanded services. They've brought in different therapeutic modalities that you're probably all familiar with. But what's interesting is that when you have a scientist together in a clinical environment with a very good clinician, you can do new things and push the envelope a little bit. So one of the big challenges that comes in a genetics clinic is the variant of unknown significance, right? You get genetic testing, there's a family, they're worried about their children, and they get this result back, and they're sort of in genetic purgatory, right? Well, there's something in, Titin. We don't know if it's pathogenic or not, but just keep in touch with us. Not very reassuring. So Anjali asked Kiran, and this is their work, what can we do for these patients? He said, well, I've got all these IPS models. Maybe we can use those in select cases to try to disentangle which variants of unknown significance are significant or not significant. And so this is what they do now. They take IPS cells, any old IPS cells, and they use genome editing to introduce a variant of unknown significance. And I have a mutant cell line. These are the same in every way except for that one change. And then they push them and differentiate them into myocytes and compare their phenotypes. and based on the phenotype, try to say, well, is this significant or not? This is cellular phenotyping. Lots of limitations. It's certainly not CLIA approved, but there are cases in which it has been useful. So I'm going to just give you two real examples. So this is a case where there was a family that presented, and one of their members had what clearly looked like a laminopathy. Bad cardiomyopathy and arrhythmias, went to transplant. And we got genetic testing, there were children involved, and the only thing they found was a variant of unknown significance in lamin A. So Kieran said, well, give me that variant, I'll put it in lamin A and see what the nuclei look like. Because the nuclei are a hallmark of a laminopathy is a blebbed and damaged nucleus. And what you see here is these are the iPS cells, the edited cells are on the bottom, the control cells are on the top, they've been pushed to myocytes. And you can see right from your chair, these are beautiful, smooth nuclei in the baseline cells. And look at these nuclei here, not so good, okay? And this is edited cells with a variant of unknown significance. So we went back to the patient, and this was a pretty rapid turnaround. This took about 6 weeks. So Kieran and Anjali went back to the patient, showed him the pictures, and said, we can't say it for sure, but I'm worried about this variant, and if your kids have this variant, we're going to keep a close eye on these kids. Okay? So this is the kind of stuff that we should be doing, trying to bring this back to our patients. Here's another example. This is a TNNT2 variant of unknown significance. So the readout here wasn't cellular nuclear structure. It was response to isoproterenol. So this is iPS cells in a dish. You give them isoproterenol, and they behave like cardiac cells. They beat faster, right? So you can see that down here. This is a patch clamping action potential. All right, so we put in the variant of unknown significance and did the same assay, and they're not responding anymore. So we said, maybe this is something we should worry a little bit about. So there are niche cases in which this might be useful, and I think scaling this is a challenge, but it sure has been fun. Okay, I told you I would come back to polygenic risk. And so the idea here is don't worry about individual mutations with strong effect. Don't even worry about individual mutations with weak effect. Take a disease in a population and regress the likelihood of that disease across every variant in the genome, whatever millions you've measured, get all the beta coefficients and assemble them into a predictive score, and then use that to predict likelihood of disease in a new patient. Turns out this works pretty well with important caveats. So here is a work done by Amit Kara in which he did this in the UK Biobank. Again, free data, download it, analyze it. And he found that you can predict, using a polygenic risk score, you can project a likelihood of coronary disease with pretty astonishing accuracy at the high end. So look at the prevalence of CAD in the people who have the highest scores, 10%. That's the same or greater than having an FH mutation, just to put it into context. So this, we think, will become useful over time. Implementing it is a challenge. And there was a commentary just yesterday, or 2 days ago now, in the New England Journal about this very technology. This has been done for many, many different traits now, and people have these scores. But figuring out how to use them is a challenge. Getting them CLIA approved is a challenge. Having the right reference populations in which to derive them is a challenge. And I'll come back to that in one moment. And here's the issue of having the right reference population. Just about everything I've talked about in this lecture so far has been derived from populations of European ancestry, because that's where the biggest data sets were first. That's clearly not how we should do things in the long run. And here's why. This is a very provocative study by Zach Cohane's group, published a few years ago now, in which he showed that there are differences in, quote, cardiomyopathy mutations in different frequencies in different populations. So variants that were said to be causal and pathogenic in Caucasian populations were actually more common in African American populations, and they're not causal. And there are many cases he identified where a family, an African American family, would come in and be counseled based on a European American data set and told they have a pathologic mutation when in fact they don't. This is wrong information. So we need diverse reference populations to do all the above: genetic testing and also polygenic risk scores. Now, happily, this will happen, but it's going to take time because there are now large-scale efforts to diversify the data sets out there. And the data are not going to be limiting. I think what's going to be limiting, again, is the phenotyping and the data analytics capacity to untangle all this. So that's where I would invest my developing expertise. Okay, so just to bring it back to where we started, where do we stand now in the value of heart failure genomics? Well, just to summarize, rare heart failure variants with strong effects, the values here. These are clinically actionable. They are the subject of genetic testing. We need to do a better job of getting these out to our practices, and that requires a change in how we think about training our cardiovascular fellows. I think there should be more genetics in the curriculum. Common heart failure variants with small effect, the value is emerging. Many of these are in regulatory regions, like we showed in the eQTLs. They can identify therapeutic targets, and polygenic risk may over time have value. And we really need more data sets in diverse populations. So with that, I'd be happy to wrap it up here and first recognize the many collaborators. This is very much a team science sport. You can't do it any other way. Too much data to get together. And I want to thank you for sitting through the lecture and for a great visit. Thank you very much.
Any questions?
Yeah, it was a really, really nice, broad-ranging talk. You alluded to the issue of scalability, and there's a very nice editorial, I think, in Science or Nature that calculated the number of genetic counselors and hours that would be required if people started showing up routinely with these panels. Who's going to be the gatekeeper in all of this? Because it isn't going to work.
No, I think what's going to happen is over time, this will become a more routine aspect of medical evaluation. And we will be the gatekeepers. We will have to speak this language and have the ability to counsel patients with guidance, with decision support, just like any other complex set of data we bring to clinical practice now. 30 years ago, or 20 years ago, you wanted to get a PCI, it was a big deal, right? Academic center, you remember this from the first stents and everything, but now it's more of a commodity thing. I think over time, this will become more of a commodity thing in the practice of medicine. But we have to do it deliberatively and make sure the trainees are ahead of the curve.
I have a question about variants of uncertain significance. Do you really need to do a whole cell model where you're actually looking at the phenotype of the cells, or can you, with computer modeling, predict the structure of the protein, how it's going to fold, and whether it's going to do its job correctly?
These are great questions. So, and it depends on how conservative you want to be and how much you believe in those algorithms. So people, there's polyphen and many other tools to predict the pathogenicity, the predicted pathogenicity of any given variant. And you can use that to inform your decisions. It's a matter of confidence and how much you believe in those algorithms. They'll get better over time, like everything else. But you're correct, that's far more scalable than an IPS model for every patient.
One more question.
That was a great talk, thanks. This is a bit of a philosophical question because this has come up recently in other areas. A lot of the genes that we are identifying through various screens, whether it be in dilated cardiomyopathy, cardio-oncology, are actually genes that have a life in cancer. And that's not surprising that genes involved in growth, might impact cardiac phenotype and, at least in cardio-oncology, things that promote survival and reduce the chemo-induced cardiomyopathy cause cancer in other tissues. And I'm just, what's your sort of idea that these genes, like the one that you identified, are you going to feel comfortable putting a patient on a medication for 20 years that may have a link to cancer? And I'm just, have you thought about how you're going to do this?
No, it's a great question. The first is so the more proximate question is this really a druggable target or pathway, MTSS1? It sounds like it might not be, but maybe it'll lead us to something that is. I think the reason why, and this is just philosophy again, there might be overlap between cancer pathways and cardiomyopathy pathways, has to do with injury response, right? So the heart responds by fetalizing itself when there's an injury, and lots of growth pathways get reactivated. Many of the same are in cancer regulation. So that might be why that's the case. But in terms of perturbing one of those genes and hoping it's going to affect heart failure, we have to use the standard tried and true approaches of trials and really look for toxicities, et cetera. There's no shortcut is the bottom line.
And what do you think it's going to do to the economic viability of doing heart failure trials if the next class of medications may have a cancer impact 10, 20 years down the road?
I think if that's really the case, it's going to be a problem.
On that excellent answer, thanks very much.